Hello everyone, I welcome you all to Biology Lessons with Kalyani, and this is the part 2 lecture on enzymes, and today we will be talking about mechanism of the enzyme action. The outlines of the today's presentation is like this. First, we will see what is the active site of the enzyme. Second, we will be discussing about the different stages or the catalytic cycle of enzyme activity. Third, we will be looking at the different factors that affect the enzyme activity and how they affect it. Fourth, we will be looking at what are enzyme inhibitors and how they impact the enzymes. Finally, we will be discussing about enzyme cofactors. We all know that enzymes are biocatalysts that lead to the conversion of the substrate into the product. An enzyme can catalyze variety of biological reaction. Now this takes place when the substrate goes and binds to a specific site of the enzyme that is called as your active site. Now active sites are basically crevices or the pockets in the enzyme where the substrate goes and binds. Now this interaction between the enzyme and the substrate is very specific in nature, almost like your lock and the key. Talking about the stages of an enzyme action or catalytic cycle of an enzyme, it has 4 different steps. So first step is the substrate, it will diffuse near the enzyme and bind to it. Second, the bound substrate will lead to the conformational change in the enzyme. In the third step, the enzyme will lead to the formation of the product, but yet the product is attached to the enzyme. Finally, in the fourth stage, enzyme as well as the products will be released in the reaction. Now let's look at it diagrammatically to make it more clear. So this is the enzyme and this is the substrate. In the first step, the substrate goes and diffuses near the enzyme. Now there is the active site which is empty, to which the substrate goes and bind, leading to the formation of the enzyme substrate complex. This binding of the substrate to the enzyme leads to a conformational change in the enzyme. Why? Because the enzyme needs to bind to the substrate very tightly so that it can alter or change the substrate and the product can be formed. In the third stage, what happens, the bonds of the substrate are broken down, it is transformed and the product is formed, but still the product is not released from the enzyme. A temporary enzyme product stage is formed. Finally, in the fourth stage, both the enzyme and the products are released in the reaction. Now this enzyme substrate complex formation is very essential for the product to be formed, and both this enzyme substrate stage and enzyme product stage, they are temporary in nature. So that's all about the catalytic stages of an enzyme, and I hope you are clear about it. Let's now look actually how the your enzyme and its active site looks like. So look, this is the end structure of the enzyme. It is a tertiary structure of the enzyme. It has many secondary structures, and the folded tertiary structure of the enzyme, it has few pockets in it, and we call such pockets as crevices. Now here the chymotrypsin enzyme image is given. Now this is the image of the active site, and you can see that the substrate, how it is binding to the active site, that it is making bonds with the residues present in the active site. That is why the binding of the substrate and the enzyme is very specific. Now you don't need for your exam purpose, these diagrams are not there, but for understanding purpose, to visualize the active site and how exactly the substrate, what is binding of the substrate to the enzyme, this is very useful. So look, this is the enzyme, and when the substrate is going and binding to it, the U-shaped active site, how it is undergoing the conformational change. So this particular image is of hexokinase, and when glucose goes and binds to the U-shaped active site, the U-shape is undergoing the conformational change, that is, it is closing in, so that the glucose is in very close proximity of the hexokinase enzyme. So let's now look at the different factors which affect the enzyme activity. Now, an enzymatic reaction, it is given by a rate, which is also called as the velocity of the reaction, and we can represent it by amount of substrate that is getting degraded with respect to time, or amount of the product that is formed with respect to time. And this velocity of the reaction is dependent upon the change in temperature, pH, substrate concentration, as well as the presence of the specific chemicals. Now, why these particular factors are responsible for changing the activity of the enzyme? To understand it, we must remember that enzymes are typically what? Made up of proteins. So proteins, when you change the temperature, pH, or the substrate, their conformation undergoes a change, and because of this there is a change in the reaction rate or the velocity of the reaction. So now let's look into the effect of temperature and the pH on the enzymes in more detail. Now enzymes generally, they function in a very narrow range of temperature and pH. Now that is, if you talk about the pH and you compare the enzyme activity at different pH, then a particular pH is there when the enzyme activity is the highest, and that pH in which maximum enzymatic activity is shown, that is called as the optimal pH. Is that clear? Now, generally it is seen that enzymes have an optimal pH in between about 5 to 7. So, if there is extreme pH in both the stages, the enzyme becomes inactive. In either very low pH or very high pH, the enzyme becomes inactive. Why? Because the structure of the enzymes or the structure of the proteins gets denatured in the extreme conditions of pH. That's why for the optimal activity of the enzymes, it is essential to maintain the pH at a particular range. Okay, let's now look at the effect of the temperature on the enzymatic activity. Now, this is the x-axis in which is showing the temperature, and this is the y-axis which is showing the enzymatic activity. Now, this is the low temperature. When you go on increasing the temperature, so the kinetic energy is getting increased. So initially you will have an increase in the enzymatic activity, and it reaches a maximum. The temperature at which the enzyme shows the maximum activity is called as the optimal temperature. At optimal temperature, enzyme shows highest activity, and the optimal temperature is always specific to a particular type of the enzyme. Now, both at low temperature and high temperature, the enzymatic activity is very less. Now, why it is less? Because enzymes, being proteins, their structure becomes inactive at both extreme low or high temperatures. Now, in case of the low temperatures, the structure is temporarily inactive, but if you increase the temperature to very high condition, then there is a denaturation in the tertiary structure and the protein activity is completely lost. Okay, so even though the rate of the reaction, it doubles if you slightly increase the temperature by 10 degrees Celsius, but if you increase it to very high temperature, a denaturation of the enzyme is seen, and hence complete loss of the activity of the enzyme is observed. Hence, it is important to maintain both the optimal temperature as well as the pH to get a optimal activity of the enzyme, and that is specific for each enzyme. I hope the effect of temperature and pH is clear for you guys. So let's now move on to the next factor, that is the effect of concentration of the substrate. So if you see, this is the x-axis, this is the y-axis, and x-axis is denoting the concentration of the substrate, and y-axis it is showing the velocity of the reaction. We already discussed velocity or the rate of the reaction is equal to amount of either the substrate degraded or the product formed, right? So look, if you initially you are increasing the substrate concentration, what is happening? The velocity is increasing, and it will increase up to a particular point in which you will get a highest activity. After that, it shows no increase in activity or the velocity of the reaction, if however more substrate you are adding, right? So why it happens like that? Because when you initially increase the substrate, more substrates go and bind to the enzyme, so there will be increase in the rate of the reaction. But when the substrate is increased beyond a point, there are very few enzyme molecules and there are no active sites remaining in which the substrate can go and bind. That's why, as there are no free enzyme molecules to bind to the additional substrate molecules, the velocity or the rate of the reaction or the enzymatic activity becomes constant and does not increase with any further increase in the substrate concentration. I hope you are clear about the effect of concentration of the substrate. Let's now look at the effect of presence of specific chemicals called as the inhibitors. Now in biological system, if you see, is very complex. There are certain chemicals which resemble in shape or structure to that of the substrate. So what happens, the binding of these chemicals that can happen to the enzyme and that can affect the enzymatic activity, right? That is, so if certain chemicals, they are going and binding to the enzyme and affecting the enzyme activity or decreasing the enzyme activity, we call such chemicals as inhibitors. And if the structure of the chemical is resembling to that of the substrate, then those inhibitors are known as competitive inhibitors, right? Not all the inhibitors are competitive inhibitors. Only those inhibitors which resemble in shape or the structure to the substrate are known as competitive inhibitors. Now, why we call them competitive inhibitors? Because there is a resemblance in the shape of the substrate. The inhibitors compete to bind with the active site. That's why they are known as competitive inhibitors. Now, due to the close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site. Consequently, the substrate cannot bind, and as a result, the enzyme action declines. So, example of it is inhibition of the succinic dehydrogenase by malonate, which closely resembles a substrate succinate in structure. So, what's happening in this example? So, here the enzyme we have taken is succinic dehydrogenase. Now, there is your the substrate which is succinate or succinic acid. Now, there is inhibitor which is malonate or malonic acid. Now, malonic acid, it is almost structurally similar to succinic acid. So what happens? Malonic acid tries to bind to the succinic dehydrogenase, thereby decreasing the enzymatic activity. Such type of inhibition is known as competitive inhibition. So here we can diagrammatically represent the same. So this is the enzyme. This is its active site. See, we have the substrate and the inhibitor. This is a typical example of your competitive inhibition. So the structure of the substrate and the inhibitor is resembling closely with respect to the active site. So what will happen? The inhibitor is going and binding to the enzyme active site. So as a result of which, it is not free for the substrate to bind. So the enzyme activity gets decreased, right? I hope that's clear now. Let us now move on to the next topic, that is cofactors. Now, what are cofactors? So if you talk about enzyme, sometimes the enzyme can have a protein part as well as a non-protein part. We already discussed enzymes are made up of protein, but sometimes in enzymes we have a non-proteinaceous part in them. That non-proteinaceous part is called as cofactor, and that is required for the enzyme activity. So if you tell the complete enzyme, we call it holozyme or holoenzyme. It is made up of the proteinaceous part, that is the apoenzyme, plus the non-proteinaceous part, that is called as a cofactor. Let's now see what are the types of the cofactors we have. So there are three types of cofactors. One is prosthetic group, another is coenzyme, third is the metal ions. So now the prosthetic groups are those kind of the organic compounds that are tightly bound to the proteinaceous part or the apoenzyme. For example, in peroxidase and catalase enzyme. So take the example of enzyme peroxidase and catalase. So you know that these enzymes, they catalyze the breakdown of hydrogen peroxide or H2O2. They convert H2O2 to water and oxygen. So if you see these enzymes, now there is a non-proteinaceous part which is attached to the enzyme and necessary for its function, and that attachment is very tight in nature. So that is heme. So heme is a prosthetic group and it is part of the active site of the enzyme. So this is very important. Prosthetic group of peroxidase and catalase is heme. Is that clear? So we saw prosthetic groups are the non-proteinaceous groups that are attached tightly to the apoenzyme. Now there can be certain organic compounds which are not associated very tightly, but they are associated loosely, and the interaction is temporary or transient in nature and occurs usually only during the catalysis. Such organic compounds are called as coenzymes. Now coenzymes serve as cofactors, and in a number of different enzyme catalyzed reactions, they are made up of basically vitamins. So vitamins are important components of most of the coenzymes. So we can take example of coenzymes can be NAD and NADP, and both of these contain the vitamin niacin. So that also is very important. The full form of NAD is nicotinamide adenine dinucleotide and P is phosphate here, right? So what are coenzymes? Coenzymes are bound loosely, not tightly as that of the prosthetic group. Coenzymes are bound loosely to the apoenzyme. There are a third category of cofactors which are metal ions, and they are also required for the enzymatic activity. Now these metal ions form coordination bond with the side chains of the active site. Okay, and also these metal ions, while the substrate goes and attaches itself to the active site, they also form the coordinate bond with the substrate. So example of such cofactor is zinc in case of the carboxypeptidase as well as carbonic anhydrase, right? When you remove these metal ions, the activity of the enzyme is removed. That shows that these cofactors play a very important role in enzymatic activity. To summarize cofactors, this is your enzyme, this is the substrate binding site, this is the cofactor binding site. This proteinaceous part of the enzyme is called as apoenzyme. Now cofactor is the non-proteinaceous part. It can be of two types, organic as well as inorganic. Now organic part can be divided further into two types, prosthetic group where the cofactor is tightly attached, and example of it is heme group in peroxidase and catalase, and second type is coenzyme which is attached loosely to the apoenzyme. Example is NAD and NADP. Another important point, mostly the coenzymes contain certain vitamins in them. Second category of the cofactories can be inorganic in nature. It is basically made up of the metallic ions. Example can be zinc as the cofactor in carbonic anhydrase and carboxypeptidase. So this cofactor goes and bind to the cofactor binding site, just generally near the active site. So this makes the enzyme functional, and this functional enzyme consisting of both cofactor and the apoenzyme is called as a hollow enzyme. So that's all about the mechanism of action of the enzymes. I hope you enjoyed the session today, and please refer to the part 1 video of the on the enzymes for understanding how enzymes catalyze the reactions in a very high rate, and understanding the basics about the enzymes. If you like the video, please subscribe to my channel, like and share. Also, if you have any queries, feel free to comment down below. Thank you everyone. Have a very good day.